Okay, I want to give a short introduction. So it's my pleasure to introduce Dr. Leah Krubitzer for today's lecture, hosted jointly by Focus in Neuroscience and the Center for Microbiome and Culture. So Dr. Krubitzer graduated with a BS in sleep pathology from Pennsylvania State University, launching right into her PhD at Vanderbilt University, where her work focused on the evolution of primates' visual cortex under the supervision of legendary Dr. John Cass. Her honors are too numerous to do full justice here, but they recently include the 2021 Power Lecture, considered an annual lecture in the Organization for Human Brain Mapping in honor of Jean Talman, President-elect of the MacArthur, and visiting scholar for the IMT School for Advanced Studies. She's now a distinguished professor at the Department of Psychology and Center for Neuroscience at UC Davis, California, with past funding from the McDonald Foundation and a current R01 on the impact of the environment on sensory motor cortex in rats. Among the many research questions that her lab focuses, the laboratory of evolutionary neurobiology, she's made sizable contributions to our understanding of the co-evolution between cortical structure and organism morphological specialization. Using a comparative approach across diverse rows of taxonomy, including prairie voles, Nile grass rats, and wild rats, the lab investigates how ecology, body morphology, and individual lifestyle shape cortical organization by manipulating sensory exposure in early developments. Among many of the innovative techniques the lab developed, one of those is the use of a microfluidic cleaning device to identify region-specific neural circuits. So without further delay, for I'm sure will be an extraordinary lecture, please join me in welcoming Dr. Leah Krubitzer to Emory.
Okay, cool. There we go. So the title of my talk is Combinatorial Creatures, Cortical Plasticity Within and Across Lifetimes. And it's really funny because this is the Center for Mind, Brain, and Culture, which is pretty cool. And I couldn't decide which talk I wanted to give. And this one I think is a good one because I end up by actually talking about human culture, even though I don't work on humans at all, and I work on a lot of different animal models, but I think they can really inform us about some really basic principles in culture. Anyway, the reason I have pictures of animals and bodies is because for so long, I just worked on brains. As a graduate student, I was in John Caz's lab. The animals got wheeled into electrophysiology room or did neuroanatomical studies on them, and you looked at, you did your experiments and bam, you have your papers and you're done. So I went to Australia to work on duckbill platypus, and I had to catch all the animals I worked on. And that was a total game changer because you realize I'm not just working on this brain thing. I'm working on this animal that's really good at what it does, really displaying adaptive behaviors for its environment. So like there's a whiteboard, Leah Krubitzer 1, duckbill platypus 250, like the number of times you went out to catch a platypus and the number of times you actually got a platypus. So I think you'll see that both the comparative work and the developmental work, it's all about brain-body environmental interactions, including affordances available to the developing brain. Okay, great. So why study the neocortex? This is a back view of a bottlenose dolphin. This is a side view of a native cat. It's an Australian marsupial. They're not they're not to scale. So this is much larger neocortex than this. And the reason I'm interested in the neocortex is the portion of the brain that's involved in complex behaviors. And I use the term complex, like for humans it includes reading, language, and species it includes cognition. It's also the portion of the neocortex, it's the portion of the brain that's changed most dramatically over the course of evolution compared to the spinal cord or the brainstem and even the dorsal thalamus. And I will admit, it's also the portion of the brain, when you open up the skull, it's like right there. And it's really easy to access and know where you're at. Okay, so big question in our laboratory is, this is a flattened view of the neocortex, rostrals to the left, medials to the top, and you're going to see a lot of these flattened views. The brain of an early mammal, maybe 200 million years ago, and the organization is deduced from comparative studies, and this is, let's say, the brain of a modern mammal. So we know from comparative studies that early mammals had a neocortex that was composed of few parts, maybe 15 cortical fields. And we know, I think people say human brains have 300 cortical fields. And the question is, how does a brain evolve from a simple form with a few parts to a complicated form with many parts? So here's the problem. You can't study the evolution of the brain directly because these types of changes take tens of thousands to millions of years. And while we can look at fossil records and have an understanding of how big a brain was, we have no idea of the internal organization of the brain and its structures and its connectivity. So there's a solution to this. I could throw up my hands and say, oh, well, I'm going to go home and have a beer and let somebody else deal with this in flies. Or, because you can't study evolution in flies. You can do a comparative approach, and a comparative approach tells us what evolution has produced. I can look at a bunch of different species at any given point in time in their life cycle and say, okay, a brain has this, a brain has this, a brain has this. And so it's saying evolution can produce cortical fields of a given size. Evolution can produce wings and hands and paws and flippers, if you're looking at external morphology. The problem is we're looking at frozen frames. So evolution is a moving picture of life, and then individuals have a moving picture of life within their own lifetime. And so anytime you're doing this comparative approach, I'm taking a snapshot, a frozen frame in time, and then I'm trying to put together the story. So while comparative studies can tell us what evolution has produced, they can't tell us how phenotypic transformations occur. Why do I have a hand like this and a platypus has a flipper? Why do mice have vibrissae and I don't? So what we do is we use the developmental approach. So we study developmental mechanisms that give rise to some aspect of the phenotype. Because evolution is actually the evolution of development mechanisms that give rise to a cortical field. So something's being tweaked in different lineages that generates those sorts of changes. So this allows me to determine how these phenotypic changes have occurred. So what am I comparing within and across lifetimes? Functional organization, cortical connections, cortical architecture, cortical field size, neural response properties, gene expression, lifestyle behavior. This is at a systems level. I can compare all different other kinds of things as well. I can compare molecular mechanisms. I can compare synaptic organization and so on. And so this is an old slide, and this is work from my lab and other laboratories. And what we found is that looking at using the comparative approach, I know that there's a constellation of cortical fields, and these cortical fields are in different colors. You don't have to know the names of them, but this is visual cortex in blue, somatosensory cortex in red, auditory cortex is in yellow. And what you can see is there's this pattern of organization that all species have. Every mammal, I should say, I'm talking about mammals, every mammal that's been examined has these cortical fields. So even before I knew, even though I don't know what the common ancestor looks like, I can say, well, here's the scenario. Either this organization was inherited from a common ancestor in thousands of lineages, one hypothesis. Hypothesis two, it wasn't inherited from a common ancestor and it independently evolved thousands of times. What's the most likely thing to happen? And so you want to look at a lot of different species to make these reliable predictions about an unknown form. And really until about 3 decades, we didn't really know much about the human brain. What we knew about the human brain came from work on other animals. So we can also make inferences about the human brain based on our comparative approach. And we were right based on our comparative approach. Okay, but here's what's really cool. Even in the absence of use, cortical fields can be identified and connections can persist. This is a beautiful example from the Bronchi laboratory. This is a blind mole rat. This is where the eyes are, and skin is grown over the eyes, and the eyes are microophthalmic. They're like the size of like a pinhead, right? What's cool though is this is rostral, this is medial, here's S1, somatosensory visual. They still have an architectonically defined V1. You can't get rid of it. They still have geniculocortical connections. You can't get rid of them, suggesting that there are huge constraints on how you can build a nervous system, right? You can't get rid of some stuff. You can make it smaller, and it's probably due to the contingent nature in which genes are deployed during development. If then. You can't just pull something out and get viability. I will say though that the visual cortex has been co-opted via changes in connectivity by the auditory system. Increases in the size of the cortical sheet is what happens when you increase the size of the cortical sheet. I can identify this is a macaque monkey that's been flattened, rostrals to the left and medials to the top. This is a mouse. These are not drawn to scale. And what I can see is here's my constellation of cortical fields, red field, blue field, green, posterior parietal cortex, little auditory cortex. I can identify them all. There are changes in their relative location with the expansion of the cortical sheet. There are changes in their relative size with an expansion of the cortical sheet. But you can still identify these fields. And of course, there's an addition of new fields in some species. And if I were to draw these to scale, that is about the size of a mouse neocortex. So if you knock this out with one of a monkey, it probably wouldn't notice anything. But I'm just saying. Okay, so what are the modifications to the neocortex that comparative studies have revealed? And we're using a variety of criteria, doing electrophysiological recordings in combination with cortical architecture, in combination with cortical connections. And these, like I said, are systems level changes that have been made to the neocortex. And there are about a handful of changes. There are a million ways in which the brain is changed. It's really just a handful, probably due to the constraints imposed on developing nervous systems. One is the change in the absolute and relative size of the cortical sheet. And these squares are meant to represent a cortical sheet. One is in sensory domain allocation. So if blue is visual, red is somatosensory, and yellow is auditory, I can see that the amount of cortex that is processing inputs from the different sensory systems can radically change. The relative size of cortical field, so I can identify a primary visual cortical field, but if I look at its size relative to the size of the cortical sheet, I can see modifications. Magnifications of I'll say behaviorally relevant sensory surfaces. If something's important, you get a lot of neural real estate. So if I'm a human, I get a lot of neural real estate for my fingertips. If I'm a duck-billed platypus, I get a lot of neural real estate for my bill. I'm not going to talk about addition of modules, number of cortical fields as I showed in the previous slide, and connections of cortical fields. And what I'm going to talk about today is sensory domain allocation, relative size of cortical fields, magnification of cortical fields, and connections of cortical fields. And how can I induce these changes in a developing animal and generate something that evolution might produce? Okay, so here we go. What factors contribute to these systems level changes? What is the time course over which these relatively large changes can occur? Can they only occur over the long time course of evolution, or can they occur actually within a lifetime? And as I said, can we induce these modifications in the developing system and generate a phenotype that is consistent with what evolution would produce? Not just a phenotype that works, but something that looks like a blind mole rat, for example. Okay, so here are the conclusions of my talk, so you can go away after you read this, because what I'm going to do is I'm going to hopefully convince you of these conclusions. And this is really important. There's no single way in which change occurs. So when I was a graduate student, I thought, what is the way in which you add a cortical field? What is the way in which you generate connectivity or difference in connections? There's not a single way. So there are a variety of different ways that this can occur. And I think there are multiple mechanisms that generate similar phenotypic outcomes. And I think a really good example is, let's say, the expansion of neocortex in a dolphin and the expansion of neocortex in humans. It's probably due to some changes in cell cycle kinetics during neurogenesis. You can increase the rate, you can increase the number of progenitor cells, which humans have done, that can then divide. You can do a whole bunch of things. Do you think the exact same things are happening in a dolphin and a human? Probably not. I think there's 180 million years of independent evolution or something like that. So there are multiple ways in which you can increase the size of a cortical sheet, for example. Any animal is a combination of different factors that contribute to the phenotype of the brain, the body, and behavior. And changes can occur over different timescales. And I'm not just talking about large changes that occur over the timescale of evolution versus, kind of little tweaks to the system that occur over a lifetime. I think you can generate really large changes within the lifetime of an individual. Okay, my outline, I'm going to give you examples from the natural world, differences in peripheral morphology. I'm going to talk about experimental manipulations in sensory receptor arrays in Armonodopus domestica. I'm going to talk about a new project called Laboratory Rats Gone Wild. And then I'm going to talk about natural variation in early experience within a population. Okay, so this is one of my favorite observations. It's what I went to Australia to do, which is to catch duck-billed platypus. And I don't know if you know about the duck-billed platypus, but it's got a bill that looks like a duck. What's really important is it closes its eyes, its ears, and its olfactory system. It doesn't use these when it's doing really important things like navigating, catching prey, and mating. On the bill, there are stripes of mechanosensory receptors that are exquisitely sensitive to touch, and stripes of electrosensory receptors that allow it to find a prey that's moving, right? And so, if you map the neocortex, if you look at the neocortex, this is a flattened view of the neocortex. This is rostral, this is medial, and I'm looking at the representation of the bill, and this is a platypunculus. And so this was actually, yes, you can make up names, it's okay. And no one, you didn't say that's not a word? Of course you're not. You can say, oh wow, I learned a new word today. Anyway, so this is just, and you're gonna see a lot of cartoon renditions, and I should explain this. This is, we advanced electrodes in hundreds of sites across the neocortex, and we looked at receptive fields of the neurons at those sites, and were they responsive to somatosensory versus visual versus auditory, and we generate these maps. And what's really cool, here's S1 in light blue. This is the representation of the body. You've got a little tail here, you've got a hind paw, forepaw. This enormous representation of the bill. It takes up about 90% of S1. And there's a distinction between mechanosensory receptors in red and electrosensory receptors in blue. There's another somatosensory representation called the rostral field. Huge representation of the bill. Huge representation of the bill in this caudal field. Auditory cortex is small and it's embedded within somatosensory cortex. And you have this little tiny pathetic little V1. They're not using it very much at all. And probably about 75 to 80% of the entire cortical sheet is devoted to processing inputs from the film. And it's not just the cortex, it's also the thalamus. And I'll just tell you this really quick story. I'm thinking, oh, there have to be thalamic changes. So I'm looking on the microscope, looking for the ventripoposterior nucleus, which is actually the nucleus associated with somatosensory processing. And I'm like, I can't find it. And you know, I don't know, if you guys are anatomists, you always like wipe off your slide, do one of these. like somehow that's gonna make a difference. It did make a difference. Because I did one of these, and I'm looking, I'm like, oh my God, I'm looking for the ventripoposterior nucleus within the ventripoposterior nucleus. It was so huge. It took over the whole dorsal thalamus with a little tiny, teeny, crappy lateral geniculate here and a little teeny, tiny, crappy auditory nucleus here. So you're seeing these different sorts of magnification along the entire neuroaxis. Okay. To what extent, so let's look at the platypus, we're thinking about the platypus. To what extent are these differences in cortex associated with genes intrinsic to the neocortex? During development, genes are deployed before any sensory input hits the developing brain. Genes associated with the development of the body, we're always looking at genes associated with cortical development, but what about the body? That is playing a huge role. And a woman, Karen Sears, does beautiful comparative work on how the body develops in different species. Epigenetic influences on body morphology, like diet, gravitational stress. You know, it's different if you're in water versus if you're in a cave or if you're flying. Sensory stimuli present throughout development. We've known this for a long time. Use of the morphology and movement options, and really important, affordances like lakes and trees and cars and whatever else there is out there in the world. Okay, so we know that genes intrinsic to the neocortex alter cortical field size. That was one of those systems level changes that I saw. This is work from the O'Leary's lab. It's been out for a while, but it's really beautiful. They've changed the expression of one of the transcription factors, EMX2. They overexpressed it. This is caudal, this is rostral, this is medial, this is V1, and it's shown with the serotonin state here. It's this size in a normal animal. And in an EMX2 overexpressed animal, you can see it's much larger. And I'm showing you it in cartoon form here, and I'm showing you that they actually measured it. But you can see it's definitely different. So all I have to do is sort of tweak the expression of a transcription factor, and I can get some of these systems level changes that I know. What about body morphology and use? This is comparative work by the Cortical lab, and there are many people like Karen Sears who have been following up on this really beautiful work. So here's my morphology of a mouse and a bat, a forepaw and a forepaw. And we say this is a wing, but this is a forepaw. And you're thinking, oh my God, there must be a million genes involved in like turning a forepaw into a wing. It turns out they're not. They're a handful they change the extent of their expression. They are turned off or amplified. And one of these is PRX1, and it's involved in the elongation of the forearm right here. So they have a really long forearm. Oh, by the way, D1, D2, D3, D4, D5, interdigit membranes, crowley, etc. And you can see here, if you compare the bat and mouse forelimb development, that the expression pattern of this gene involved in this is very different in the bat. And there are a number of others. And I love this picture because it's just so beautiful. And I should say that the wing also has specialized hairs that are exquisitely sensitive to small changes in air pressure, which is a really good thing if you're self-propelled flight. Okay. So this is a nice comparison, not my own. If you look at the organization of S1 and the forelimb representation in a mouse versus a bat. This is a microcropping bat. And what's really nice about this comparison is that the brains are about the same size. And what we see is this huge representation of the forelimb in a bat. Is this due to changes in genes that are deployed during development? Is it due to changes in genes that are constructing the body? Is it due to the use of that particular morphology? Or is it due to some combination of all of those things? And I would argue it's probably due to some combination of all those things. But it could, in fact, just be due to changes in peripheral morphology and use. I mean, we don't know that, and people have not really studied that. Okay, so what I would argue, and Ken Catania has given, if you guys have read his papers, beautiful. If you haven't, you should definitely read them. And he's given us many beautiful examples of this magnification in a Starnos mole, where these follicles are very important for everyday behavior, and you have an expansion of that representation of somatosensory cortex. Really nice stuff in the naked mole rat, where the lower incisors can be moved independently, and they use this for identifying objects, navigating other things. I talked about the build of platypus. But if you look at the supralaryngeal tract of humans, you have your specializations of the musculature, specializations of the tongue, and we have what's called a Broca's area. But I would contend that this is just an expansion of this representation of somatosensory cortex, motor cortex, and premotor cortex, alterations in connectivity. So while it is a human specialization, it's still following the same rules of construction of other species. It's not doing something out of the ordinary. Okay, so second part, experimental manipulations of sensory receptor arrays. So what we're doing is, so the idea is, what happens if I change the ratio of sensory inputs during development? And so with and this, like I said, is experimental. It's like taking a sledgehammer to the system, but you want to see what's going to happen. Because you'd say, okay, with the duck-billed platypus and some of these other species, you're radically changing the ratio of tactile and electrosensory inputs coming into the developing nervous system compared to visual and auditory, which are very, very small. And so what we've done is we've said, okay, let's take a visual animal. This is our Monodelphis domestica. It's a South American marsupial. It's small, nice. This is rostral, this is medial. And if I look at, this is I can't remember what stain it is, but this is primary visual cortex, somatosensory cortex, and auditory cortex. And we've done electrophysiological recordings across the cortical sheet and identified that this is functionally V1, and visual cortex actually expands out here. And what we've done is we've removed the eyes, and this is what it looks like at P4, which we do this. Prior to the retinal ganglion cell axons innervating the thalamus, they're just at the optic chiasm. Also prior to thalamocortical afferents reaching the developing cortex. And it sounds a little gruesome, but it's not, because the eyes are just this little thin layer of cells. And so you just remove them in about 5 minutes, and then you let the animal grow up, and you see what happens. And so here's what happens. So if you look at the size, so there's no change in the overall size of the cortical sheet, but if you look at the relative size of V1, you see that it's really, really small, and S1 has gotten bigger. This is similar to the O'Leary lab, where they've changed by overexpressing EMX2, and they made it larger, and they did the same thing backwards, where they can make it smaller, and we've done it by simply changing the ratio of incoming sensory input. But I can't get rid of V1. I can identify it architectonically, again. So this is similar to what evolution has produced. And if I do electrophysiological recordings, and each one of these is a recording site, so the blue neurons respond to visual stimulation, the red somatosensory stimulation, and the green auditory stimulation. Here's my normal animal, somatosensory cortex, visual cortex, auditory cortex. If I look at my bilateral nucleate, what I find is that all of what would be visual cortex is now taken over by the somatosensory and the auditory system. I can still identify and architectonically define V1. And so, if I look at the receptive field properties in this reorganized V1, I find that they're all in the snout, the face, the periphery, and the head. Very few on the lower limbs, and very few on both. And so if I look at this, I'm saying I have this really reorganized V1 where I've magnified what's now a very important behaviorally relevant body part, which is, and I'm going to show you, it's basically the whiskers. But here's what's cool. We also saw changes in S1. So this is work by Deepa Murthy, who was a graduate student in my laboratory. She recorded from neurons in the Brissy representation of these early blind animals. And what she found is that there are changes in the receptive field size. So in early blind animals, you had very small receptive fields, and in compared to sighted animals. And she looked at the discriminability of neurons in S1. How well can I detect the stimulation of one whisker versus the surrounding whiskers? And they have a high discriminability index compared to the sighted controls. And she found a lot of other cool things, but I don't have time for that. Okay, connections. These are super old studies. These are the basic connections of V1 in a monodelphis. V1 is getting input from other visual fields and from the visual thalamus, and S1, A1 getting input from their respective nuclei of the thalamus. And if you look at early blind animals, it's maintaining the basic pattern of connectivity with changes in density, but it's now getting input from auditory nuclei of the thalamus and cortex, and somatosensory nuclei of the thalamus and cortex. And really, this is normal somatosensory connections, just an overview, but we're also seeing smaller changes, but they're still there. So oftentimes when we're doing these sorts of experiments, we only look at the target system, but it's like throwing a rock in a pond where the largest perturbation is going to be where the rock goes in, and then you get this sort of ripple effect. So you're getting changes throughout the neocortex. Okay, so what about behavior? So I've shown you these changes in connectivity, I've shown you differences in response property, I've shown you this magnification of behavioral relevant body parts. What does it mean for behavior? And I'm interested in behavior because behavior is the target of selection and external phenotype. Genes co-vary with the target of selection, but they are not, and cortical field size varies with the target of selection, and as well as connections, but they are not the actual target. They are dragged along with the target. And so these are, I have to say, these are the hardest experiments, these behavioral experiments, because I was doing neuroanatomy and neurophysiology for years, And then I started doing behavior, and I realized, I talked to some of the people today, like I was getting trained, I was like really well trained, and the monodelphins were not trained at all. So it took a long time. You're like, oh my God, look at me, I'm getting up at 5 o'clock in the morning to get crickets of a certain size, I get on my bike, I start the trials every day, I've done that for 3 or 4 months, and they'll do like 2 trials. But I was like, my alarm was set at the same time, but I had this routine, I'd get up and eat my oatmeal, it was kind of crazy. All right, so anyway, what I want to show you, and I think this is really cool, and I hope you can see it, and I don't think I can do it, I think I have to do it here. So, the reds are the nucleates and the blue are sided, and I will say this is a different experiment where we actually reared the animals in an enriched environment. But what I want you to notice, here's their nest box, and they're down here right around. If you didn't know that these animals are blind, you couldn't tell the difference. Oops, hold on. Okay, is it going? Yeah. Okay, they're not this fast. It's sped up. But here's our guy. Here's our bilateral nucleate. He's getting little nesting materials. He's putting in his little prehensile tail, which is so cool. And he's navigating this environment. And I should say, she changed out every few days. She changed the configuration of everything so that it wasn't like a spatial memory task. And he's getting stuff, and you can see him go right up to his thing, right, without any errors. It was really, truly amazing. They had the same activity level. Activity was spread across the cycle, the diurnal nocturnal cycle, but it was pretty cool. Okay, and I'm not going to show you this, but she did tactile discrimination tasks. Early blind animals have better tactile discrimination, and it is mediated by the whiskers, because if you whisker trim, the behavior falls apart. Better tactile discrimination than sighted controls. So this is work, relatively recent work by a former graduate student of Mackenzie, England, and this is a variable lateral run walking task. And so basically, there's a sighted animal, here's a blind animal. You have this ladder rung suspended across the floor, high up, but it's in plexiglass, so you get the illusion that you're going to fall, but you're not going to fall flat on the floor. And so you have to train the animals. There's a cage they start from, and their home cage is here. And they really want to get to their home cage. And so you start with the rungs are equidistant, and they do this really fast. It's a natural behavior. And then what you do is once they know what they're supposed to do, then you randomly change the spacing of those rungs. And so I'm going to show you, this is awesome. So this is a sighted animal. Because you can do stats and everything, but when you see this, it's like, oh my God, it's like a result. Okay, so this is a sighted animal. And you can see them going, and miss is when their paws go through, right? The high paw or forepaw, and there's different ways that you can show that. This is an early blind animal, and unfortunately it's at a different speed, but you can see he's doing pretty well. With a couple little errors, but not many errors. Now check this out. This is the early blind animal who had his whiskers trimmed. Totally falls apart. I know everybody says, oh, the whiskers grow back. He finds his love of his life and they get married and they're happy ever after. But you get the idea. It's such a powerful result. So we're showing in cortex there are changes in the representation of the whiskers, receptor field properties are changed, and now we're showing, I think this is doing something for behavior, right? Which is super cool. Okay, and if you look at the early blind animals outperform these, and this is total error is lower than in sighted animals, and if you whisker trim, they both fall apart. When you whisker trim, they sort of look like normal animals. And I'll just say, we're also doing what's called deep lab cut. I think you guys do behavior, you all know what that is. What's really cool is that you can look at postural changes. So what animals look like, they're really careful. So you can look at how high they're lifting their foot, how far they're lifting their foot. And so it's like when you turn off the lights, you're walking, and all of a sudden the lights go off, and you're doing this. And that's kind of what they're doing. They're really, really conservative in how they approach this, and they're nose tapping a lot more. And there's tons of this data, but I'm not going to show it to you. Reaching and grasping. What is the contribution of the spare sensory systems? So this is now a new task where the animals have to, there's a little cricket, yes, the cricket, placed here, and they have to reach and grasp. And there are a couple different wells here, and they have to reach and grasp, and you see how accurately they can use. And this is super hard to train these animals to do this, but Carlos has done it. And then he's done something amazing. He's trimmed the whiskers in the same animal, let the whiskers go back, and then he's made them anosmic to say, okay, what's the contribution? And so here, I'll just show you. This is what a miss looks like, and this is what a hit looks like. And this is a sighted animal. So here's look at this. He totally misses it. So even, and he gets it. So here's good news. Here are the results. So this is a light condition when the lights are on, and this is when the lights are off. When the lights are on, sighted animals do better than the blind animals. Good news. Vision contributes to this task. In the dark, they do the same. If you trim the whiskers, they both get crappy, significantly more crappy. That's a scientific term. You can use it in your results. If you make them anosmic, they get really bad. But here's what I want you to know. Look at the variability in the anosmia. When you take away their sense of smell, their world falls apart. Now, we've also done saline controls, and that doesn't happen, so it's not just a procedure. Their world totally falls apart. But then they get better. Then I'm not going to show that, but then they get better. And then we did some deep lab cut on this, and I think you guys know what that looks like. You can see the animal. This is an old one, so we've come a long way since there. And this it's cool because it can actually tell you what is their snout elevation, shoulder elevation. Do they have the same kinematics once their whiskers are trimmed? Are they doing it the same way? What is their compensatory behavior to get this test done? Okay, but as I said, that was sort of like taking a sledgehammer to the system. Oh, one last thing. So we just started these experiments, and I'm not going to show you any data because we're still working on it. But what we want to do is, everything I'm showing you is correlative, that yeah, these brain changes are really doing something. And I think it's strong correlative behavior. What we're going to do is we implanted cannulas in these guys, in visual cortex, and we're going to do them in somatosensory cortex, and we're going to inject muscimol, and we're going to deactivate these regions during the performance of lateral rung and Michigan grasping, and see what happens, and see if there's a direct causal effect of this reorganized visual cortex and somatosensory cortex. But I said, that was like taking a sledgehammer system. This is a new, so this is going to be me because as soon as I'm done here, I'm going to swig some Delcium. And then they're going to take me out for dinner tonight, and I'm going to have some alcohol, and it's going to be interesting. Anyway, we finally got this project funded. We've been working on it for a long time, like a side project, and we call it Laboratory Rats Gone Wild. And we were lucky. This is done by Fernando Gomez, who practices in my lab, and Chris Breezy, whose background is engineering. And I'm going to show you some subsequent slides, and I'm 100% certain you're going to be able to figure out which slides he generated. So the question is, can animals really exhibit species-specific behavior in an environment like this? So we're lucky because we have these outdoor field enclosures that are 3,000 times larger than the laboratory cage. And we have all kinds of different textures, logs, we put natural grasses in there and hay, different places where they can get water, they have nesting boxes, and we change this configuration once a week. So even though it's not a dynamic environment, it's a changing environment. And they live out there, so temperatures range from 43 degrees to 12 degrees, humidity 4 to 100%, so it rains. There was actually an earthquake out there, larger than the one in New York, which, anyway, I was actually in an airport in New York when they had that earthquake, and it sucked away my will to live. It was so awful. 18 hours to get from A to B. Okay, and they have wild predator cues, wild turkeys, raptors. The predators can't get at them, but we also have little critters climbing in their cage, and we feed them and water them and do the whole thing. Oh, what I should say, my bad. The mothers are bred out there, and they give birth out there. And so we're using these Norwegian browns. And then we do the same thing with the laboratory animals. So it's these two parallel experiments. Okay, and so let me just show you. I mean, there's you guys get what I'm going to show you here. So these animals can do stuff that laboratory animals can't do. So they're vertically climbing, they're using their tails, they're jumping, and they're doing all kinds of stuff, right? And so you don't have those movement options in a field pen, in a laboratory. And you don't have these affordances. Like they actually, during the summer, we have a nest box, but they will build several nests on their own. Even the way they use their trunks to get water. And as I said, they're doing some vertical climbing, they're using their tails to brace themselves. And so what we did is, this is intracortical microstimulation. I think I showed this a little bit at that neural control movement. And so basically you're sticking an electrode in the brain, and at each site, and these are anesthetized animals, you're putting in a small current and you're seeing, okay, what body parts move. And if it's a single color, here is the jaw, it means that you had a small jaw movement. If it's multicolored, like yellow, light green, dark green, it means you had multiple joints that were activated during that stimulation period. And so this is sort of complex multi-joint movements, this is simple movements. And so we did in these developing animals, and we're still collecting this data, but I think if you really look at it, you can see the difference, and we're starting to quantify it. Here's a post-natal age 40 Labrador rat, post-natal age 40 field pen rat, and you can see there are a lot more multi-joint movements. You have a large representation of the hind limb, forelimb. If you look at post-natal age 60, this actually gets even more complex, where you're having these trunk hind limb movements. I wish I put in here an animal where we had a lot of tail hind limb movements. So you're getting these differences. So the map is more extensive at P40. There are more multi-joint movements at post-natal age 60, and more extensive hind limb representation, and more forelimb synergies at post-natal age 60. Fernando is also doing these neurodevelopmental studies to see when they start to generate coordinated movements, when they get into locomote, and it's interesting because the behavior's a little bit variable. If you look at the rats, lab rats, it's really clear when things happen, and it's pretty tight distribution. And in the rats, it was a little funky. So then what we did is we broke it out by season, and it turns out that variability, they behave very differently in the summer than they do in the winter versus the fall versus the winter. So what we did, I think what I want to show you here is if we look at synergies, when we can evoke movements from multiple limbs. The green is field pen and the blue is laboratory animals, so forelimb, hind limb, many more. Forelimb, hind limb, tail, we've never evoked those in the laboratory rats, and we evoke these synergies that are commonly used in the field pen animals. Forelimb and tail, so you get the idea that we're getting really big differences based on commonly used synergies. And this is just a lateral walking task, and what it shows is that early in development, the field pen animals have many fewer errors, but then it kind of goes away. Okay. Engineer, not engineer. I'm like, Chris, give me one of your newer slides. So he gave me one of his newer slides, but this is really cool. So he's looking at the somatosensory cortex. And what he did is really awesome. He quantified the statistics of the textures in the field pens and in the lab, so that we can then regenerate those when we're doing electrophysiological recordings in the somatosensory cortex. And what he found, so lab environment has constrained range of textures. So if you look at, this is a lab rat in red, and this is a field pen, and if you look at the number of textures, you can see it's pretty constrained versus this really broad distribution of textures out in the field pen. Not surprising. Record from neurons in the paw, and diversity representation S1 and examine texture, specificity, and firing rate. Okay, again, one of his newer slides. I love it though, it's great. And basically we have this rotating drum where we have 3 different ones that have a range of textures, and we're either stimulating the forepaw or the diversity in the lab animals versus the field pen animals. And this is just an example of what you get. This is a strong excitatory response in the forepaw of a field pen. On is the green and off is the red, and you can see you can do this really well. We have inhibitory responses, but these are the response profiles. And one idea was that, are these neurons going to be really broadly, are they going to have broad tuning curves for texture in the field pen versus the laboratory? And it seems like they do. So it's responding to a wide range of textures compared to the more restrained number of responses for a given texture in laboratory rats. And this is just what we're showing. It's only for the forepaw. It's not for the diversity. Only for the forepaw representation, neurons have a higher firing rate in field pen versus laboratory animals, and neurons in the forepaw representation of field pen rats respond to a wider range of textures. But here's the big question. So this is all cool. This is beautiful data. It's nice, and it's nice too because it's like an experiment where I don't have any skin in the game. I kind of don't care what comes out, right? It's not like I have this huge hypothesis that I want to prove. What's really nice though is it's nice for me to have this hypothesis because almost everything you look at is different. But the big question is, can rearing in a highly variable environment with a large number of affordances alter the ability of the animal to meet a wider range of sensory motor challenges? So, like, if you think about lab animals, and we've all had IOPA come through, if your temperature deviates from 72, you're like, okay, you've got to work this out, and it can't go more than 4 degrees. The lights are on and off at the same time every day. They get their cages changed at the same time every week. There's no variability in that environment. That curve is so tight. Whereas if you look at the field pen animals, there is a huge amount of variability in their experience. And so I'm going to show you this because I'm really excited. Because this is like fresh off the press. And so it looks like a crappy setup because we haven't actually built something yet. We're just sort of testing something. So here's a video. He said, okay, how about if we have animals climb up a rope? Now, the field pen rats, they vertically climb, but they've never climbed up a rope. And of course, the lab animals have never climbed up a rope. And so what I'm going to show you here, I'm going to show you individually, and I'm going to show you together. It's so cool. So this is the time it takes them to, this is a plexiglass open space that they do not want to be in, right? They want out and they want to go up to the experimenter's hand or a box up here. It takes laboratory animals 5 minutes to even approach the rope thinking, hey, I can get out of here. It takes these guys, the field pen animals, 20 seconds. And then I want you to look at their climbing behavior when they do that. It's crazy. Okay. So this is the lab animal. And he's fast forwarded. 2 minutes, 3 minutes, 4 minutes, 5 minutes. A lesser man would have given up, I'll tell you. I would have. And it's taken him, and you'll see this. You can't see it down here, but you'll see it up here. And we're going to do some deep lab cut on this too. And you can see the use of his tail. He's kind of almost hopping up with his hind limbs. See what he's doing? It's taking him forever. Poor guy. Okay. And he eventually does it. Okay. Now check this out. Okay. Down here. 23 seconds. Now watch this. Like, tell me. And look what he's doing with his hind limbs. Look what he's doing with his tail. Look how he's climbing. And look how fast he does that. Like, you know, when you get these results, I know I sound like I'm bragging, but if you don't love your own data, then get out of the field. But you can actually see that. And of course we're gonna quantify that. And we literally did that like 2 weeks ago. And it's like, and then the thing with this is, and we're gonna build a real apparatus, you gotta do it the first time. It's a first off thing. Like, okay, that's one part of the data. Then you can do it more times and say, how long does it take until the lab rats get good? How good do they get? How do they compare? We can do a deep lab cut. You can see they're using their tail differently. You can see they're using their hind limbs really differently. The lab rat seems to be almost hopping and using his hind limbs as a whole, where the other guy's like doing this kind of stuff. So it's as if those brain changes in motor cortex really have some impact on the behavior of the animal. Okay. Final set of experiments, and then I'm done. Okay, so this is now a natural. This is looking at natural differences in brain condition. These are prairie voles. Prairie voles are monogamous and biparental, so that means both parents rear the young. And what we've done is we're just looking at, this is in conjunction with Karen Bales' lab, looking at tactile contact. So there's a normal distribution of prairie voles, and some parents have a lot of tactile contact, some have little contact, and some there's an average. So we're looking at these two ends of this distribution. And high contact offspring become high contact parents. And this is just showing you red is high contact, that there is much more, significantly more tactile contact, mainly with this perioral region of the mouth. When you cross foster, so this is cool. When you cross foster, if you switch vole infants on the day of birth, the cross-fostered offspring adapts to the rearing style of the parent. So it's sort of like social transmission of a rearing style. And we did the tactile contact because we can actually measure it. It's not like, is it a good mother, is it a bad mother? I mean, it's just looking at contact. And so these are experiments by Adele Selke, who was a postmortem, and they're really hard experiments because she made injections of anatomical tracers in the perioral region of S1 and matched them. She matched them for the size of the injection. You have to. You can't inject a forelimb of one and the perioral region of another. You can't have a huge injection of one and a small injection. This is great. So you say, oh, you kind of look similar. These small dots are neurons that are connecting to this perioral region. But you say, well, look, I'm looking at, I see differences in density. I see almost no connections and lots of connections. And she did quantify the density of connections in both species, in both groups. And then if you look at contralateral connections, you're like, yeah, you know, there are differences. The point is, these aren't huge differences, they're very subtle differences. And this is a very subtle experiment, but imagine these sorts of small, subtle differences across the entire neuroaxis. And that gives you a lot of degrees of freedom for really big changes. Okay, so I'm going to conclude here. What factors contribute to the cortical phenotype? Genes. And genes can change cortical sheet size, cortical field size, connections, cortical morphology, we know that. Genes that are generating cellular mechanisms involved in plasticity, so it's not generating phenotypes, it's generating flexibility, allow the environment to actually change some of these same things. So there isn't a single way, there's a combination of ways in which you can do this. I gave you an example to build a platypus, I talked about tongue, larynx, lips like a human being, but what about things like social learning, language, and culture? I would argue that these are simply complex patterns of physical stimuli impinging on the developing nervous system throughout the life of an individual and its species over time. So a mother's love is temperature, it's cadence of the voice, it's visual stimulation, it's this packet of physical energy that's impinging on its developing nervous system. It's a ratio, like we did with our blind opossums. Okay, so this is sort of a little chart of human evolution. At the top is genes and morphology. At the bottom is artifacts, social contexts, and inventions. And so if you look here, beautiful work by Sergio, it'll come to me, Almeida. He's shown that the modern hand of humans has been around for about 1.8 million years. And our big old huge human brains have been around for about 500,000 years. So the hardware has been there, right? But yet, you know, we had personal ornaments 300 years ago, sophisticated blades. The Industrial Revolution occurred 250 years ago, and our behavior here is way different than our behavior here, even though we had the hardware. Earliest anatomically modern humans have been around for about 300,000 years, which means no change in DNA sequence. And if you believe that all behaviors generated by the brain, and I do, then these sorts of behavioral changes, from sophisticated blades to Industrial Revolution to man on the moon, machine learning, human genome sequence, and daily prolonged interactions with machines and computers, means our brains have changed. And they could have changed in a lot of subtle ways within the lifetime of an individual. Because if I was Leah Krubitzer, born 300,000 years ago, it wouldn't be like I'm the same Leah Krubitzer and I'm wearing like a different set of clothing, like you know, Willow Flintstone necklaces and shit. I would be a different person, and I would be a different person if I were born 5 years later or 5 years earlier. I'd be a different person if I were born 2 years ago versus now. And I would contend, so within a lifetime. But we also have second by second by second changes as well in synaptic efficacy. So your brains are different now than they were when you walked in the room, and they're different now compared to now, not by huge amounts, but this continually changes. And this is work by Reardon et al. It's a comparative study between macaque monkeys and humans. And what they did is they looked at the portions of the brains that have expanded over the course of evolution. And those are like posterior parietal cortex, frontal cortex, intertemporal cortex. And they're also looking at, compared to macaque monkeys, which brain areas are developing later. And that's also posterior parietal cortex, frontal cortex, intertemporal cortex. And I would contend these areas are being built during development based on context. They're very different than primary somatosensory cortex or visual cortex or auditory cortex or sensory regions. And then, and this allows culture and technology to impact them directly within the life of an individual. Okay, so I'll end by saying we are combinatorial creatures constructed by genes, bodies, behaviors, and the environment. That humans have evolved an extraordinary capacity to construct our neocortex based over the course of a prolonged infancy and childhood, allowing for rapid phenotypic change even within a single generation. We have built a remarkable fluid brain-body interface with the environment, such that tools and machines, and this I think is really important, can extend our embodiment in peripersonal space and expand the loop between our brains, our bodies, and the world. And this has made us into unique biohybrid creatures, for better or for worse. Our technology has been incorporated into who we are, and it certainly is impacting how our brains develop. And our brains adapt and bootstrap themselves with the technologies they've given rise to, and with whom our futures are increasingly entwined. So even though I don't work on humans, I have something to say about humans. And I have something to say about everything. If you Krubitzers, we have an opinion about everything. And I would say I'm not opinionated, I'm just right. But I think it's important because there's this, like, what makes humans different, and we're looking for these specific things. And I'm not suggesting there aren't specific things, but there could be these general things too, that we have this cortex that can be assigned during development, and we have a lot of it based on context, and that can be very, very adaptive. And these are some of my sources of funding. This is my lab. I don't have my undergraduates here, and I have to get a picture with them because they are like the powerhouse of the laboratory as well. And thanks for coming and letting me give a talk, and I'm done.
Questions?
Yes?
I don't want to ask those. So we were talking about in this talk mostly in the context of mammals. Do do you have any familiarity with work in other vertebrate, and like, do you have any thoughts about how?
I have some thoughts, and people ask about birds because birds are, I hate to say smart, but I'll say they're complex because complex is a better word. And I would, since we see this in all mammals, and remember, what I'm looking at is a marsupial, and that, you know, their ancestors branched off really early in evolution. So I would say this ability probably predates mammals, and it's going to be different structures in a bird. But birds are different though too. So I don't know if anyone's done these studies, but sure, I'll speculate and say, why wouldn't? Why not?
To what extent do you think the fields are the same? Like, do you think that
Well, like, you know, birds presumably don't have a cortex, but they have a dorsoventricular ridge. So, you know, I mean, so I'm talking about a general principle of regions of the brain to be flexible, and maybe some more so and others less so. So you can have stereotypic behavior in birds, which you do, that you can't knock, you know, you can muck around with them a little bit, but you can't get rid of them. But that birds are capable of change, right? So, and if their behavior is generated by the nervous system, then there have to be these mechanisms in place. But I'm not a bird person. You know, it's unfortunate because we're your bird people, your mammal people. You know, I'm like anything from the head up, I'm good. Don't talk about anything from here. You know, we specialize. We can't help ourselves. We have to.
Thanks for the great talk. My question is thinking about the synergies in the muscle movements that you saw, and it seems like that there needed to be a diversity of affordances for that to emerge. Do you think there are synergies in the percepts, particularly the tactile percepts? That is it not just that you need rough and smooth, but you need rough and smooth to be experienced close in time?
Yeah, I think that's probably, because remember too, you know, when we do our experiments and we're looking at, let's say, are you talking about texture right now? Texture discrimination. I mean, animals held in place, and you're doing this. And when does that really happen in life? No, I mean, understanding texture is a learning experience, right? When it happens, where it happens, what I did before, what happened afterwards. So if that's your question, then I think that's going to play a huge role in how you perceive textures and what textures mean to you. Does that make sense? Or am I just rambling? Or is that the pseudo talking?
I can't answer about the pseudo so I would imagine that a potential experiment that one could do in the lab is to raise your animals normally and then have another group that gets in some way the same range of textures that is available to those out in the pen. However, it's always like one day at a time.
Yes.
On day one, you get smooth. On day two, you get really rough. On day three,
Right. I get you. Okay. I get your question. That's a good idea. We haven't done that, but that's a really good. And you know what? That's an easy idea. Relatively easy to implement, I think. That's a good idea. Okay. That's what
The fact that you thought it was a good idea probably is the pseudo talk.
I'm returning. You said I gave a good talk. That's a good idea. That's a good talk. That's a good idea. Yeah, we can go back to it. Oh.
Actually, before then, let me ask a question too. As you focus on cortical area and its role in evolution, but I'm also wondering about functional connectivity and potentially its focus and whether you've looked, say, anatomically at that. I'm thinking of, just in terms of heritability, oftentimes within families, you see certain behaviors reemerge over and over again, which are not just due to sensation, but some loop between sensation and action, which seems like it would involve a whole circuit. And whether you think about evolution, because it's acting or selecting based on behavior, on actually sort of internal circuitry.
Right. So we are, the study with the Rascon Wild also involves looking at connections. Intrinsic, so for example, if I'm looking at what are the connections of a forelimb, some sort of forelimb wrist movement, what are the connections? Is it only connected to other forelimb representations, or is it also connected to hind limb and tail? Is it also connected to, is this what you're sort of getting at?
Yeah.
So we're definitely doing that. Very important. And I think it's also important to do it, you want to look at within population variability as well, right? then we're also going to say, okay, so how is it connected to somatosensory cortex? I kind of am expecting, the first I'm really expecting, and you know what, we've done some preliminary data, and I think that's what we're going to get. But you also want to see how it's connected to somatosensory cortex. And it sort of gets at that guy's question too. So, you know, that really brilliant idea. What are the differences in connections there? And also, I think also important is corticospinal. Because corticospinal connections, they're not complete until like postnatal day 21 in rats. So I think that's gonna change as well. But we are definitely looking at that. But again, so I'm looking at what are the differences between field pen rats and laboratory rats, but what are the differences between field pen rats within this population and within lab rats within this population? We always want to assume there's just like, we're looking at the normal, but can we quantify that variability? Just like we did in the voles. And it's hard though.
Selectively breed, right?
Well, okay, we've already done, you know what? That was an NSF grant from like 10 years ago. So you can do, let's do some reaching and grasping in rats, or let's do the rats that do really well on the ladder rung or really well on the rope, and then selectively breed them and see if you can actually like totally like enhance that within a population. And my guess is probably, because that's the way evolution works, right? You can have normal distributions and you're really good at one task and this animal's not so great at doing some sort of thing. And why can't you have the population drift if the environment and the circumstances change, where now this tail end becomes what's adaptive.
Yeah, that's great. It's related to it's my comments. I really like the rope climbing
I know.
thing. I love it. And because I'm very interested in individual differences in movement, how they generalize across different types of movements based on people's history. So we always talk about dancers and football players walking differently in Emory. You could probably create that by having different types of environments. Right? One where you have to jump more, and one where you have to, I don't know, crawl more, or something like that. Which then you would probably not have normal distribution. You have two bimodal distribution.
Right.
Which would be great to see how they perform different tasks. Now, the reason in our work we are interested in rehabilitation, people always ask us, if I do a lot of tennis or do a lot of Tai Chi, am I going to like survive a stroke? You guys can do that experiment, right? You can look at how life experiences like that allow a sort of, I guess, resilience and movement in the face of injury.
Yeah, but maybe it's not so much about training on a particular task, because does training on one task generalize to everything else? It might be more about during development and throughout the lifetime presenting a variety of things that will make that brain. So the idea is like, will I recover better from stroke if I do Tai Chi? Well, if you've been doing Tai Chi and dancing and playing a piano and doing a whole bunch of stuff throughout your lifetime, maybe that's the argument you make. Is that like,
yeah, absolutely.
You want to tell these people who have a stroke, if you do this, it's going to be better. But you're like, you should have started when you were 2.
Yeah, we see that's protected, but we can't go back and collect data on people's life histories, whereas you have a very nice way of controlling their life history.
Well, there's tons of experiments we could do. That's what I love about this. It's like being in graduate school again. Where the world's your oyster. Like anything's like, why don't you do this? And I do say good idea, because they are good ideas, and they're really easy to implement.
All right, I'll come back to grad
Lab animal versus wild animal comparison seems really important, and part of the reason I say that is because studies that have compared human brains with like our closest living primate relatives, like chimpanzees, are comparing humans like us with captive chimpanzees, and so you have me worried that if we had instead compared them with wild chimpanzees, our results may be very different. And so, are you aware of anybody looking at the difference between wild and captive primate species, like just going beyond rodents?
Well, here's the deal. I'm not. And the thing is, that's not, because I get that. It's not like, oh, everybody has to work on something that's in the wild. You can't. You simply can't do that. And I mean, the reason we actually started this was years ago, I think in 2012, we were comparing visual cortex in squirrels, and rats. And we were catching squirrels, and our rats were lab reared. And it's like, well, this is not fair, right? We should look at what real wild caught. And we did actually catch wild rats who, I'm here to tell you, are mean. Really mean. And we saw differences. And I mean, in terms of the primates, you guys have a primate center in which animals are reared in natural social conditions. We have a primate center in which animals are reared in natural social conditions. So you could do this. The problem with trying to do it in primates is that it's really expensive and tough. And so we're doing it in rats because it's doable, and these questions can generalize. But you can't worry about, well, I did most of my research on caged animals, caged monkeys, all kinds of caged monkeys, you know, and some wild caught. But I'm not going to say that's invalid. But I would say, yeah, there's probably differences, right? I mean, because we all think about that. And when I was writing this NIH grant, which got rejected like a billion times, I love the criticisms. Well, you're the only person who can do it. And it's like, well, yeah, that's innovation number one. You know, and it's sort of like, no, I'm not trying to tell people. I mean, almost everything we know about the brain has come from work on caged animals. And I'm not going to argue that it's all wrong. But there's subtleties and there's differences. And so it's time to move forward. And I think it's really great because there are many, many more people who are looking at natural behavior. So Chris Neal in his mice are looking at more naturalistic behaviors. And I talk to people here, you're looking at more naturalistic behaviors. Even if the animals are lab reared, it's not like I'm going to sit and be head fixed and have stuff presented to me. I'm going to be allowed to explore the environment, make my own choices in this task. And so I think we're moving towards that. And you know, you got to work with what you got. And with the chimpanzee stuff and the great apes, I mean, that's a treasure trove. You get whatever you can get out of that is extraordinary.
Well, it's interesting to think about what even would be the natural environment for a human, right? So, is this it? Maybe
That's
not.
right.
Maybe
I think it
it's
was.
hunting
Well,
together.
Chris Breezy, the engineer, I'm right though. You guys could tell which slides were his. I mean, they're hilarious. It's like, okay, Chris, what does this mean? I want to think of the reference that if you look at human perception in urban dwellers versus non-urban dwellers, it's different in terms of how they detect edges and stuff like that because the urban world is about lots of edges. And so, yeah, I mean, and there's another great one. I teach this lecture on, I teach a seminar on evolution and development, and there's a great section on domestication. And a big question is, are we self-domesticated? And with that domestication comes kind of bad consequences, like your brain gets smaller and all these sorts of things. So yeah, I mean, go get a wild-caught human and I'll look at it, right? Because it's hard to know with culture whether we're self-domesticated, what is it actually wild. But I would say it's not fair because evolution just is. And so it's not wrong to be on your cell phones every day. It's not wrong to be entwined with technology. It's not wrong that we're going to, you know, if the technology fails, we're going to fail. This is just what's happened during the course of evolution. And so, okay, this is the sweet type talking. I got to stop. No, stop.
Yes, I was just at the spectrum between in early development, cortical patterning, so the degree of sort of conserved patterning of the cortex before founding innovation, and the amount of cortical specification that's actually induced by thalamus. And so even with the nucleation, I presume there's still genicular innovation actually happening, which is in part specifying a visual field. So in the absence of that and being able to redirect visual fields, where do you sort of see the distinction between cortical patterning and sort of early specification of cortical structure?
Where do I see the distinction?
Or the sort of interplay of which patterns?
I don't know if I know what your question is. So I'm doing this before, while there's still early patterning of the cortex before the thalamus hits the cortex. And so your question is...
How flexible is the cortex plant now in terms of being able to...
I think it's probably really flexible. And I get your question. So here's the deal. We've started doing P12 enucleations. This is after the thalamus has innervated the cortex, and we don't see that same flexibility in terms of representation. We're only starting to look at connectivity. So yeah, you got to do this kind of early. But it's still different in P12 than it is in like P36 or adults. In adults, you don't see these sorts of massive changes.
Hi, great talk. In your discussion with Lena, actually, you kind of got into the idea that early life experience is kind of what matters in kind of patterning of the brain. And I'm just curious, if you thought about, if you swapped those field mice and those lab mice at some point, and kind of what properties, can you speculate what properties you think, would be able to be switched with those like lab mice going into the field, have these broader tuning properties? I would imagine they could climb the rope better, but can you just comment on early life experience versus recent experience?
Yeah, I will. And the thing is, what I didn't talk about is the mothers. So it used to be that we got pregnant mothers that were shipped to us, but that was just like such a pain in the ass and didn't work out. However, we did do this with a number of mothers that were actually reared in the lab that were then out in the field pens. And so it's the same idea, right? And we didn't do enough, we didn't examine their brains enough before we started having them, now it's all just field pen generated animals, but it's a really good question. So I'll tell you what happens when you put those lab mothers, pregnant mothers into the field pens. They do really well. But we didn't do the kind of measures that we did. And what I might expect, it's a good question. I mean, if you're going to have changes in connectivity, which I think we see, then that's kind of early in development. Let's say you have intrinsic changes in connections of M1 and then extrinsic changes. If you have changes in cortical spinal stuff, that you can't undo that, right? But there's still room for flexibility. And we know that from all Mike Merznick's work and John Coz's work that, you guys wouldn't be here if we weren't capable, if our brains weren't capable of change, right? But what you're getting at is changes in synaptic efficacy. So would you see the same huge effect? I don't know. But there are ways to address this by simply taking those lab rats and putting them in the field pen and vice versa.
All right, last question here. That was actually my question. I thought of a second one, which is about I know with the mother fostering and the licking stuff, there's been a lot of epigenetic studies with that.
Yes.
And I was wondering if you're interested in looking at kind of any epigenetic or intergenerational effects with this.
I think it's a great question, and people have asked that question before, how long would this persist?
Yeah.
Even if you subtract the environment, because you got to remember, you can say, well, this persists over generations, but if the animals are in the same environment, then is it just a masquerade of evolution, but it's really not? And that's like $1 million question. At what point do these changes get incorporated into the genome and evolve? And I don't know, because this was I just gave a talk on this not that long ago, and it was sort of this cost benefit, having it being genetically determined versus let's be really flexible. And I don't know the answer to that question, but this is something that, and I'm not the person to do it, right? I don't do epigenetics, although I do collaborate with Danielle Stolzenberg, who's really interested in epigenetics and maternal behavior. But I think it's a great question. I don't know the answer to it.
All right, with that, let's give